Welcome to this lesson on transcription. Transcription is one of the most fundamental processes in molecular biology. It's the mechanism by which cells copy genetic information from DNA into RNA. This process is essential for gene expression, allowing cells to use the instructions stored in DNA to make proteins. Think of transcription as creating a working copy of a recipe from a master cookbook that must stay protected. So what exactly is transcription? Transcription is the process of copying a segment of DNA into a complementary RNA molecule. The term comes from the idea of transcribing or copying information from one format to another. During transcription, the cell reads a specific gene on the DNA molecule and creates an RNA copy of that gene. This RNA copy, called messenger RNA or mRNA, carries the genetic instructions to other parts of the cell where proteins will be made. You might wonder why cells need transcription at all. Why not just use DNA directly? There are two main reasons. First, to protect DNA, the original DNA, a molecule contains all of the cell's genetic information and must be kept safe in the nucleus. It's too valuable to risk damage during protein synthesis. Second, to create copies. By making disposable RNA templates, the cell can produce multiple copies of a gene's instructions without touching the original DNA. These RNA molecules can be used for protein synthesis, and then broken down and recycled when they're no longer needed. Transcription happens in three distinct stages. Stage 1 is initiation. During initiation, an enzyme called RNA polymerase binds to a special region on the DNA called the promoter. This is where transcription begins. Stage 2 is elongation. In this stage, the RNA polymerase moves along the DNA, reading the genetic code and building the RNA strand as it goes. The RNA molecule grows longer and longer. Stage 3 is termination. When the RNA polymerase reaches the end of the gene, it detaches from the DNA, and the newly made RNA molecule is released. The transcription process is now complete. The key enzyme that makes transcription possible is RNA polymerase. This remarkable molecular machine performs two critical functions. First, it unwinds the DNA double helix, separating the two strands so that one strand can be read. Second, it synthesizes the complementary RNA strand by adding RNA nucleotides one by one, following the base pairing rules. RNA polymerase is like a molecular copying machine that moves along the DNA, reading the genetic code and building an RNA copy simultaneously. To understand how RNA polymerase copies DNA, you need to know the base pairing rules. DNA contains 4 nucleotide bases: adenine, thymine, guanine, and cytosine. During transcription, these bases pair with RNA bases according to specific rules. Adenine in DNA pairs with uracil in RNA, thymine in DNA pairs with adenine in RNA, guanine in DNA pairs with cytosine in RNA, and cytosine in DNA pairs with guanine in RNA. Notice that RNA uses uracil instead of thymine. This is one of the key differences between DNA and RNA. Let's look at a concrete example. Imagine we have a DNA template strand with the sequence thymine, adenine, cytosine, guanine, guanine, adenine. When RNA polymerase transcribes this sequence, it creates a complementary mRNA strand. Thymine pairs with adenine, so we get adenine in the RNA. Adenine pairs with uracil, so we get uracil. Cytosine pairs with guanine, so we get guanine. Guanine pairs with cytosine, giving us cytosine. The next guanine gives us another cytosine. And finally, adenine gives us uracil. The resulting mRNA sequence is adenine, uracil, guanine, cytosine, cytosine, uracil. This demonstrates how the genetic code is faithfully copied from DNA to RNA. Let's review the key takeaways from this lesson. First, transcription is the process that copies DNA into RNA, creating a working copy of genetic information. Second, RNA polymerase is the essential enzyme that catalyzes transcription, unwinding DNA and synthesizing RNA. Third, Remember that uracil replaces thymine in RNA, which is a fundamental difference between DNA and RNA molecules. And finally, messenger RNA carries the genetic information from the nucleus to the ribosomes, where it will be used to make proteins. Understanding transcription is crucial because it's the first step in gene expression, the process by which genetic information becomes functional proteins in the cell.
If you like this video, hit that like button and don't forget to subscribe. Visit codelucky.com for more such useful content.